Hello and welcome to my channel Sam Biology Class. Friends, today we will discuss about pyrosequencing. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Pyrosequencing is a method of DNA sequencing. It is based on the principle of sequencing by synthesis. This technique detects pyrophosphate, which is released when a nucleotide is added during DNA synthesis. This pyrophosphate triggers a cascade of reactions involving different enzymes such as ATP sulfurylase and luciferase to produce detectable light signal. The intensity of the signal is proportional to the number of bases that are added during DNA synthesis. By doing so, this method provides a real-time and quantitative information about the DNA sequence. The principle of pyrosequencing was first described by Bertil Peterson, Matthias Uhlen and Paul Nyren in 1993. Instead of using fluorescently labeled nucleotides, pyrosequencing monitors the process of DNA synthesis by detecting the byproducts of nucleotide incorporation. What is the byproduct of nucleotide incorporation? It is pyrophosphate. We know that DNA polymerase catalyzes the formation of a phosphodiester bond between the 3' hydroxyl group of the growing DNA strand and the 5' phosphate group of the incoming nucleotide. During the formation of phosphodiester bond, the 3' hydroxyl group performs a nucleophilic attack on the alpha phosphate, as a result of which phosphodiester bond is formed between these two. At the same time, these beta and gamma phosphate groups are cleaved up together as a pyrophosphate. This is the phosphodiester bond which links adjacent nucleotides. When one nucleotide is added to the DNA strand, one molecule of pyrophosphate is released as the byproduct. The number of pyrophosphates released during DNA synthesis is equal to the number of nucleotides that are incorporated. Pyrosequencing detects the release of this pyrophosphate. So by adding different suitable nucleotides one after another using DNA polymerase and then detecting the release of pyrophosphate after each addition, pyrosequencing is able to identify the nucleotides and the order in which they are incorporated to the 3' hydroxyl group of the growing DNA strand. This is the principle of pyrosequencing. Based on this, DNA fragments can be sequenced. Key steps of pyrosequencing. First step is sample preparation. The DNA of interest is extracted from the sample by different methods such as enzyme lysis and mechanical disruption. The extracted DNA is purified and then fragmented into small pieces by using restriction enzymes or mechanical methods. Second step is amplification by polymerase chain reaction. In this step, the purified DNA fragments are amplified by polymerase chain reaction, which generates multiple copies of each fragment. During amplification, one DNA strand is amplified by using biotinylated primer that labels one DNA strand with biotin. For example, this is a double-stranded DNA fragment which is to be sequenced. For amplification, one of the strands is amplified by using forward primer and the other strand is amplified by using reverse primer which is biotinylated. After several rounds, we obtain very large number of DNA strands, some of which are labeled with biotin. Out of these, only biotin labeled DNA strands which are of same type are used for pyrosequencing. These biotin labeled DNA strands are isolated from the PCR product by using streptavidin coated magnetic beads. Streptavidin coated magnetic beads. Streptavidin is a protein purified from the bacterium Streptomyces abedini. Streptomyces abedini. Streptavidin forms a strong and stable complex with biotin. Because of this binding, streptavidin coated magnetic beads are used to immobilize biotin labeled DNA strands on bead surface. Third step is sequencing reaction. It begins with the incorporation of different deoxynucleotides in a predetermined order. There are two types of pyrosequencing. First one is solid phase pyrosequencing and second one is liquid phase pyrosequencing. In solid phase pyrosequencing, DNA templates are immobilized on a solid support such as streptavidin coated magnetic beads or porosilica beads. Streptavidin coated beads immobilize biotin labeled DNA templates on surface. After immobilization of DNA templates, the beads are loaded onto the wells of a flow cell. What is flow cell? A flow cell is a picotited plate containing numerous wells. Each well is loaded with a streptavidin coated bead which has DNA templates on surface. The flow cells serve as a microreactor. It allows for the precise and sequential addition of enzymes and substrates during sequencing. It also allows for the capture of light signal which is generated when a complementary nucleotide is added to the 3' hydroxyl group of the growing DNA strand. For sequencing, we require 4 deoxynucleoside triphosphates such as DTTP, DCTP, DGTP and DATP alpha-S. DATP alpha-S means deoxyadenosine alpha-thiotriphosphate. It is an analog of DATP. In place of DATP, we are using DATP alpha-S because the substrate can be efficiently incorporated by DNA polymerase. But the substrate will not be recognized by luciferase. If in place of DATP alpha-S we will use DATP, then DATP will be recognized by this enzyme, and in this case luciferin will be oxidized to oxyluciferin that will generate a false light signal. In order to prevent false light signal, in place of DATP, we are using DATP alpha-S. We also require adenosine 5-phosphosulfate APS. Different enzymes that are required for pyrosequencing are DNA polymerase, sulfurylase, and luciferase. In this figure, this is the DNA template and this is the sequencing primer. For solid phase pyrosequencing, the magnetic bead along with the attached DNA template, the sequencing primer, DNA polymerase, ATP sulfurylase and luciferase are all taken in the wells of the flow cell. For pyrosequencing, nucleotides are added one at a time in a predetermined order. Only the nucleotides which are complementary to the corresponding nucleotides of the DNA template will be successfully incorporated. Only when a nucleotide is incorporated, a phosphodiester bond is formed and one molecule of pyrophosphate is released. This figure illustrates the principles of pyrosequencing. This is one magnetic bead which has one single-stranded DNA fragment on its surface. Actually, a bead can immobilize millions of DNA fragments on its surface. But in this figure, for simplification, only one DNA fragment is shown. This fragment is the DNA template whose sequence has to be determined. For this, the template is hybridized with a sequencing primer which will provide 3' hydroxyl group to which deoxynucleotides are added by DNA polymerase. In this figure, this is the order in which we will add various deoxynucleotides for the synthesis of the complementary strand. Here you see that on the DNA template, the first nitrogenous base is cytosine. So according to this order, first of all we will add DTTP. This nucleotide will not be incorporated because thymine is not complementary to cytosine. Next we will add DGTP. It will be successfully incorporated because guanine is complementary to cytosine. So here between these two nucleotides, phosphodiester bond will be formed and one molecule of pyrophosphate will be released as the byproduct. Next we will add DATP alpha-S. This nucleotide will be recognized by DNA polymerase and will be incorporated. This is because adenine is complementary to thymine. So here again a phosphodiester bond is formed and one molecule of pyrophosphate will be released. Next we will add DTTP. This nucleotide will not be incorporated because thymine is not complementary to cytosine. This will be followed by the addition of the remaining deoxynucleotides in the assigned order. Out of these, only 8 nucleotides, those are complementary to the corresponding nucleotides of the template strand, will be successfully incorporated. In solid phase pyrosequencing, after addition of each nucleotide, the wells are washed in order to remove unincorporated nucleotides and other reagents. This is necessary to prevent false light signal. Incorporation of each nucleotide generates one molecule of pyrophosphate. So in this example, total 8 pyrophosphates will be released. Each pyrophosphate will react with adenosine 5-phosphosulfate to form ATP in the presence of enzyme sulfurylase. This ATP then reacts with luciferin in the presence of another enzyme called luciferase to yield oxyluciferin. As oxyluciferin returns to the ground state, it emits light. In pyrosequencing, we know the order in which various nucleotides have been incorporated. We also know which nucleotides have been incorporated. Based on that, we can determine the sequence of the newly synthesized DNA strand. So the sequence of the newly synthesized DNA strand will be 5' end, then G, after that A, then again G, followed by T, followed by C, then again C, then T, and then G. So this is the sequence of the newly synthesized DNA strand from which the sequence of the template strand can be easily determined. This is because the sequence of the template strand is complementary to the sequence of the new strand. So in this way, in pyrosequencing, by immobilizing a large number of DNA fragments on magnetic beads, genomic sample can be sequenced within short time. In pyrosequencing, the intensity of the light signal is detected by a sensitive camera. The sequencing reaction generates a pyrogram, which is a graphical representation of the light signals. It has been plotted by taking intensity of the light signal on y-axis and nucleotides added on x-axis. In a pyrogram, the sequence of these peaks of light corresponds to the sequence of nucleotides that are added. In pyrogram, the height of the light peaks is equal to the number of nucleotides that are incorporated during the synthesis of the complementary DNA strand. In a pyrogram, the sequence of these peaks of light corresponds to the sequence of nucleotides that are added. These light signals are analyzed to determine the sequence of nucleotides of various DNA fragments. After this, multiple DNA fragments are assembled by using specific bioinformatic tools to generate the complete DNA sequence. Friends, this is all about pyrosequencing. For more updates, subscribe my YouTube channel, Sam Biology Class.